Okay, it's lesson 2 of respiration, Krebs cycle, and oxidative phosphorylation. Outcomes for today: explain how hydrogen ions moved into membrane space, that's for A and A star. Secondly, describing how the Krebs cycle, which is for B grade, and then thirdly, recalling how ATP is made. If we want to look at the syllabus spec, that's here, so you can have a quick read of that in your time and see how that all links into the spec. You'll notice some of the words from the last lesson, so ATP and NAD, we've got a new one here, look, FAD coming into it. ATP is chemiosmosis, all things that we saw last lesson, phosphorylation and so on, all of that revisited. Okay, start us off from last lesson. These are things really I think you should be able to do. So recalling what chemiosmosis is, obviously that was quite challenging from last lesson, and what glycolysis is, and being able to draw out the process of glycolysis. And this is quite typical coming up in the exam papers, what are the products of glycolysis. Just to remind you, in terms of chemiosmosis, if you recall, we were looking at a diagram like this of ATP synthase. So you've got your intermembrane space here, slightly different name there, but it's still the intermembrane space. You've got the matrix here, and obviously this area here is the membrane. You've got ATP synthase with the gamma part around the top, which is rotating. As the hydrogen ions pass through, and for every 3 hydrogen ions that pass through there, you're able to turn 1 ADP into 1 ATP. That was chemiosmosis. If you remember, I said that was the end of the process, and that we were then going to work back to the start of the process through glycolysis and so on. Quick video. This is the one that should have shown last time but didn't show for one reason or another, some technical difficulties.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient.
If you remember, we call that an electrochemical gradient, where you've got the greater number of hydrogen ions on this side of the membrane compared to this. Just for future note, these extra proteins that you mentioned in the membrane, they're the electron transport chain, which you see later on. Well, later on today when we get through the link reaction and Krebs cycle.
The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
Or not. Okay, so that's what we were looking at. Could you from that now in your mind explain what chemiosmosis is? Hopefully you've got a fair idea of the keywords, you could structure an answer to that. We'll have a quick look now at glycolysis, what the process is, what the products are, because actually they're the typical exam questions. So you looked at this last lesson. This is glycolysis. Key things: number of carbons. Here, 6 carbon, 6 carbon, 6 carbon, 3 carbon, and then the input of ATP here and here. We didn't mention these last week. This is glucose kinase and phosphofructokinase. They're both the enzymes that drive these reactions here. And remembering the bis instead of bi, hexose bisphosphate, splits into 2 triose phosphates, and then goes through a series of reactions where intermediate compounds are formed, ultimately resulting in 2 lots of pyruvate. ATP produced, so 4 there, which gives you a net gain of 2, and as well, very very important, your 2 lots of NAD are made as well. Okay, so back to what we're looking at today. How's hydrogen moved into the membrane space, describing the Krebs cycle, and then recalling how ATP is made. If you remember, we looked at this last lesson. So the things that we dealt with last week were chemiosmosis and glycolysis. We're now going to fill in these gaps here, what goes on here. Alright, it is complex, but if you bear in mind throughout the whole of this that chemiosmosis requires hydrogen ions, it requires that electrochemical gradient in the mitochondria, then you'll be fine, because you'll be able to playing on words, you'll be able to link this whole system together, so you can see where things are going and why things are happening, and that'll help you understand it in the long term. Okay, so we talk about glycolysis, you need to know that through and through. Now then, the link reaction, it's literally as it says, it's a link. It joins the Krebs cycle to glycolysis. Basically, it's a dehydrogenation, a decarboxylation of pyruvate. You can work those out. Dehydrogenation, taking away hydrogen. Decarboxylation, taking away carbon dioxide. And ultimately, what you're left with is a 2-carbon compound called acetyl coenzyme A. It looks something like this. So your pyruvate goes in here, 3 carbons. It's decarboxylated, dehydrogenated, and what you're left with is this acetyl coenzyme A. Coenzyme, it's an enzyme. It helps this reaction. It joins to this 2-carbon compound and forms acetyl coenzyme A, and ultimately that then feeds into the Krebs cycle, which I should be able to show you now. So just down here, so you can see it looks like an enzyme there, and it's chemically unchanged at the end of the reaction, so it doesn't change. It literally just drops off those 2 carbons into the Krebs cycle.